Good morning, ladies and gentlemen. We're delighted to welcome you to the Spectradyne webinar that's going to be held today and delivered by Dr. Jean-Luc Fraikin, the CEO of the company. Dr. Fraikin always gives a fantastic webinar presentation, very engaging. And today the talk is going to focus on EVs in complex biofluids with the Spectradyne particle analyzer. So without any further ado, ladies and gentlemen, we'll turn it over to Jean-Luc to initiate the webinar. Welcome, Dr. Fraikin.
Thanks, Anil. Thanks, everybody, for joining us today. Like Anil said, the focus of today's webinar is to talk about measuring EVs, extracellular vesicles, in complex biofluids. And in particular, I'll show you some examples of immunostaining EVs directly in plasma and how that can be done accurately and quite easily on the ARC particle analyzer. So if you don't know Spectradyne, our company is really dedicated towards making accurate nanoparticle analysis technology. And we have a mission of reshaping the discovery, development, and production of modern therapeutics, so mostly nanomedicines, right? EVs in particular are an important component of that, obviously, because their applications are, because they show so much potential, right? So they are involved in many different areas of fundamental biology, and as such are being developed for use as therapeutics, as biomarkers, and even just to understand the fundamental biology. At the size scale, the particle size and concentration, though, are super important because they dictate the physical behavior, the nature of these materials. So they impact directly the dose, the biodistribution of a therapeutic, and ultimately the safety and efficacy of a drug, or how they perform in a diagnostic assay also. In this kind of scenario, in this kind of application, it's important also to be able to measure the cargo that each EV is carrying, because that's really related to the dose, also their potency, and also their safety and efficacy. So Spectradyne really focuses on measuring three basic things, right? Our technology is pretty simple, actually. Just measures the size, the concentration, and the amount of cargo on these EVs, these nanoparticle-based therapeutics or biomarkers. These are important for all areas to quantify the critical quality attributes for all areas of EV research. One example I like to use to highlight this is if you're trying to compare two different preparations or formulations of EVs, call them A and B. A lot of times the way this is done is by applying them to a test system like tissue culture media, where those formulations A and B affect different changes in the test system, in the model, right? So maybe it's tissue culture media, maybe it's an animal model, but the idea is you apply these formulations to the test system, and then you measure the outcome. So maybe that's a flow cytometry assay or some other metric to determine the outcome, right? In this experiment, though, you would never, ever consider doing this without first controlling for the input, the dose of the material applied to the test system, right? If you put in 10 times as much of formulation B as formulation A, it's natural that you're going to expect a different outcome from those two formulations, right? So measuring the size, concentration, of payload, which are all related to dose, right, is really just critical for doing basic good science. There are a lot of technologies out there for measuring the outcome of this experiment, like flow cytometry we mentioned, right? And those are even in approved diagnostics now, which is really cool. But there is a severe lack of technologies that are appropriate for measuring these nanoparticles, the input to this experiment. And so, but there are many available. Okay, so let's talk about those. There's dynamic light scattering, is very common, mostly in LNP applications. There are different varieties of surface imaging-based particle analysis tools. Some of them are newer than others. There's nanoparticle tracking analysis, which is probably the most ubiquitous technique for measuring EVs because it was there when the field was started. And increasingly, flow cytometry is being applied towards measuring EVs directly in samples also. So a lot of tools out there. There are some commonalities, though, among all these tools. 100% of them are optical techniques, right? So that means they use light to infer the properties of the particles, including their size. And as such, they're indirectly measuring the particles, right? You have to make assumptions about the particles in order to extract those conclusions. Also, because we're using light to measure nanoparticles, these methods are fundamentally limited at the nanoscale. And that's because the particles are smaller than light. So if you're using light scattering, then you're heavily dependent on the optical properties of the particles in order to get the signal to begin with, let alone to make measurements about them. So in some cases, it's possible to infer or calculate a particle size from the light that it scatters. But in order to do that, you have to know the refractive index among other variables of each particle that's being measured. And those kinds of assumptions cannot be made in heterogeneous samples. So today we're talking about heterogeneous samples. It's impossible to make a blanket statement about the optical, properties of the particles in a serum sample, for example, right? You've got protein aggregates, vesicles, lipoprotein particles as well. They're all mixed in there. They all have different refractive indices. So you can't use light scattering to accurately measure the size distribution and therefore the concentration of these particles in these samples. I would like to pick on the wrong word, but I'd like to show an example of that using flow cytometry, because this is increasingly used for nanoparticle analysis and for vesicles as well. So this figure that I'm showing here is from a paper in 2019. It's from Beckman-Coulter, actually, when they developed the Cytoflex. And what it shows here are measurements of 3 different kinds of materials at various sizes. So all the green dots are polystyrene beads. All the yellow dots are viruses. All the blue dots are silica particles. And what they did was, and so the size of those particles has been determined with some other method, right? And they measured the scattering intensity from these particles on their flow cytometer. And the point of this paper is to show that, is to make a model of the refractive index of nanoparticles. And that's what these streamlines are. They show lines of constant refractive index according to their model that they developed. And you can see that generally the polystyrene falls along a single streamline, which makes sense because they all have the same refractive index. Viruses are similar and silica is similar too, right? What I want to do with this figure is imagine a real world scenario where you have a mixture of different materials in your sample. So for example, let's imagine mixing this polystyrene 125 nanometer particle, the HSV1, which is around 150 nanometers, and the silica 200 nanometer particles together, okay? And so we know, because we know the size of these particles, that the true size distribution looks like this. The polystyrenes are the smallest, the viruses are in the middle, and then the silica particles are the largest ones in the sample, right? So this is what the actual particle size distribution looks like. And you could measure the concentration, across any one of these ranges, right? The problem is when you use a blanket assumption of refractive index to infer the particle size of particles in this sample. So if we do that, the polystyrene particles actually scatter more strongly than any other particle in the sample. And so they give the biggest scattering signal, and we would infer that they would be the largest particles in the sample this way. The silica, even though it's the biggest particle in the sample, scatters in the middle. And so we would put that peak in the middle of the distribution. And then the virus particles, very weakly scattering, kind of like EVs, would be measured would be inferred to have the smallest size if you just use light scattering. And so essentially, my point here is that if you use light scattering to measure a particle size distribution, then if you have a heterogeneous sample, it's not going to come out right, okay? And you're going to get the wrong size distribution and concentration, right, as a result. So light scattering based methods yield inaccurate size distributions when the sample is heterogeneous, okay? So there is still a critical need for direct and accurate measurements of EV size, concentration, and cargo, even though all the flow cytometry companies are piling in with more and more sensitive flow cytometers. They're not accurately measuring the size distribution of these particles, okay? This is where Spectradyne comes in. So this is our ARC particle analyzer, which is the instrument I'll be talking about today. It makes just very simple and direct, accurate measurements of EVs or any nanoparticle for size, concentration, and fluorescence. Okay, we start at about 50 nanometers and go up from there. Really importantly, there are no assumptions required for the accuracy of this technology. So its accuracy is independent of the refractive index of the particles, the polydispersity of the sample in terms of size, or the heterogeneity of the sample. And I'll show you plenty of examples like that today. And it's very fast and easy to use. So if you're used to using flow cytometry where you have to align the beam or calibrate the intensity or clean out your flow cell, you don't have to do any of that on the ARC. It's totally automated or not required at all. On the flip side, though, the ARC is compatible with all the flow protocols and reagents that you're used to using. So you don't need customized microarrays, to do a custom assay. Just like on a flow cytometer, you use your combination of antibodies or reagents that you like. So this is the ARC. Today, I'll show you lots of examples of data measured or collected from the ARC. Let me explain how it works really briefly if you're new to this. So at Spectradyne, we use an electrical method to count and size particles. And the magic happens inside this microfluidic cartridge. It's about 1/2 inch on the side. You pipette your sample into this reservoir, and then inside the cartridge, what we do is flow the sample so that one particle at a time flows through a sensing constriction, okay? And when a single particle goes through here, it generates an electrical signal that tells us two things. One is how big the particle is, so that's related to the amplitude of the signal. And secondly, how fast it went through the constriction. And so the speed of the particle tells us the flow rate of the sample. So we're measuring the sample volume by this flow rate, every particle we detect, right? So we're measuring the volume of the sample and we're counting particles. That's the most direct method of measuring concentration, right? We're counting particles in a known volume. So that's how the concentration and the sizing work. And this technology has been, we call it microfluidic resistive pulse sensing, or MRPS. It's been on the market from Spectradyne for about 10 years in the form of the NCS1. This instrument's been globally deployed, and there's a bunch of publications out there you can go and find. And more recently now, in the ARC particle analyzer, we're coupling the same detection method with single particle fluorescence. Okay, so we excite each particle with a laser as it passes through the constriction simultaneously with the passage, right? And we detect the fluorescence emitted from that particle. And we can do that in up to 3 different color channels. So in the ARC, we're measuring size and concentration electrically. So this is a non-optical technique, right? Means that we don't suffer the same limitations as all those optical techniques. And we're also measuring, just like on a flow cytometer, single particle fluorescence in up to 3 color channels. So this is quantitative measurements, and I'll show you examples of all that today. In fact, the electrical method here has been a gold standard for decades for measuring bigger particles in the form of the Coulter counter, right? But Spectradyne's innovation has been how to scale that down to the nanoscale in a way that's practical and usable for really complex samples like I'll show you today. So some of those engineered features inside the microfluidics are shown here. And I want to draw your attention to the one shown in panel A. This is a filter structure that's built into every one of our cartridges. So it's not a typical membrane filter that you would think of when you go to filter your tissue culture media, for example. This is really just a network of small channels through which the sample must pass to be detected. And it just captures particles that are too big to fit through the constriction. So, in more simplistic implementations of RPS, the whole sample flows onto the nanoconstriction, the sensing constriction, and that causes major clogging problems, right? Spectradyne's innovation has been to embed these filters into each cartridge so that you can analyze complex samples that contain large particles much more easily and much more practically, and still measure the nanoparticles in those samples. As I mentioned, it's very fast and easy to use. So there's no cleaning required. There's no calibration required for the instrument for size, concentration, or fluorescence intensity. And there's no optical alignment either. So if you're used to flow cytometry, where you have to make sure that your beam's aligned with the sample stream inside your flow cell. You don't have to do any of that. We use an automated image recognition system to do that. And in fact, it's adaptive. So during the course of a measurement, it's always checking to make sure that the alignment is optimal. And that allows us to be able to deliver pre-calibration for brightness as well as size and concentration. One last point here is that because we're in microfluidics, we benefit from the small volumes, and only 3 microliters of your sample are required for analysis on the ARC. Okay, so let's get into some data. So first, I'll show you an example that is just illustratory, really, or illustrative. It's just a super mix of polystyrene beads, okay? And there are 6 components in this sample, and diameters range from 100 to 290 nanometers. There's a bunch of different fluorochromes in this sample. Some are non-fluorescent, and then there's different varieties of fluorochromes. They have overlapping size distributions, and each one is added in at equal concentration, right? So just to give you a sense for what the raw data looks like, this is an example from measuring this sample. So in blue is the electrical signal, the MRPS signal, and it's showing detection of 2 particles in this very fast time frame, right? This is a scale in microseconds. So 2 particles detected here. The first one also generates a fluorescent signal in all 3 of our fluorescence channels, okay? So these are shown in green, yellow, and red. And so this is a fluorescent particle here. And then this particle, the second one, is a non-fluorescent particle, and it does not generate any fluorescence in our detectors, right? So we're just doing the electrical detection of the particles for this one. Right, so when we measure the entire sample this way, this is the result that we get. So what I'm showing you here is now a few different particle size distributions, where on the y-axis is particle concentration, just in regular particles per mil units, right? And then on the x-axis is the true particle diameter. So this diameter is not inferred from the light scattering. It's just we've physically measured the size of each particle as it goes through the constriction electrically, right? So there are a few different things to notice here. First of all, we can resolve all the different size components. So these are the 100s, these are the 180s in the sample, there are 200s, and there's a broad distribution of 250s, and then these are the 290s, okay? And each distribution is color-coded to show what population in the sample is being shown. So in blue, we're showing the particle size distribution of all the particles in the sample. In green, which you can't quite see, are just the green fluorescent particles. The yellow is the size distribution of just the yellow fluorescent positive particles, etc. Now, we can resolve all the different sizes, but we can't see the difference between the 180s just using size here. Because they overlap in size, right? But when we look at multi-parametric scatter plots for these measurements, which is shown here, we can readily identify all and quantify each component of this mixture. So what I'm showing you in this big panel here is a 2D dot plot, a scatter plot, which you'd be familiar from flow cytometry if you're used to using that. It shows a dot for every particle that's been measured, okay? And we're showing the particle diameter in true nanometer units, not inferred from light scatter along the x-axis, and the brightness of that particle on the y-axis. So here now you can see the difference between the 180 YG particles and the 180 Y particles because they have different fluorescence intensities in our green channel, right? And so you can use the different color combinations to identify all the components of this mixture this way. All right, so this is just a bead example, and you don't have to take my word for it. Anyway, we have independent validation, including winning the CYTO Innovation Technology Showcase last year at the CYTO annual meeting. This is a flow cytometry technology conference every year. And we have customers in 3 main applications. So nanomedicine, meaning mostly LNPs. We also do a lot of virus and gene therapy applications. And here today we're talking about measuring extracellular vesicles on the ARC. I'll just draw your attention to this publication. The reason I like this one is from the NIH, Dr. Jones and Dr. Welsh from the NCI. reason I like this publication is because they really combine two orthogonal methods. So they use conventional flow cytometry, it's a Cytec instrument, with the Spectradyne arc particle analyzer to generate new information, right? So they combine those two different views of what's in the sample to measure the refractive index of individual EVs this way. And it can't be done with one perspective alone, right? So this is a cool paper. I encourage you to go check it out. Let's get into some arc measurements of EVs now, generated by us. So the first example I'll show you is measurement of recombinant extracellular vesicle. It's available from a company in the UK called Vesicles. So if you don't know these guys, go check them out. They have a lot of cool products for fluorescence-based measurements of EVs and other nanomaterials. So this particular product is a recombinant EV that expresses a fusion protein between CD63 and GFP. So what that means, so CD63 is a tetraspanin, right? It's one of the common EV tetraspanins. It's a transmembrane protein, and some of them in this sample then are conjugated to GFP also. So this is a natively fluorescent particle this way. When we measure the unstained sample, so just the product itself on the arc, this is this particle size distribution we measure. So it's got a pretty broad size distribution. When we look at the fluorescence, it's a little more interesting, shown in the bottom, top right here. So this scatter plot is showing brightness on the y-axis in the green channel versus brightness in our PE, a yellow channel, on the x-axis. So this is the dimension we're going to use to do immunostaining of these particles in a second. So we have detection limits. They're quite good relative to flow cytometer standards. So, and we could talk about these units in a Q&A at the end if you like. By the way, we'll have a Q&A session at the end of the webinar. So either put your questions in the chat or you can raise your hand later. We can talk directly. So we have detection limits, obviously. So anything that is above this horizontal line is considered fluorescent positive in the green channel, right? Anything that's to the right of this vertical line is considered fluorescent positive in our yellow channel. So this sample natively expresses GFP, and so we expect to see this nice distribution of fluorescent green particles in the sample before modifying it in any way. The story gets more interesting, though, when we stain it with an antibody for CD63. So first, the size distribution after staining with the anti-CD63, the PE conjugated anti-CD63 antibody looks about the same. That's normal. We expect that the particle size doesn't change very much when you tack on a few different antibodies on the outside. So that's fine. But the fluorescence profile of the sample changes radically. So that's what's shown up here. Now in blue, so what we see is that pretty much anything that is GFP positive, so it has the GFP on it, also picked up the PE stain from the anti-CD63 staining. And that makes perfect sense, right? Because the GFP is fused to CD63. So we should expect that anything that has the GFP there also has the CD63 and is double positive for that. We don't get a lot of particles that are GFP only that are not also expressing CD63. And then there's also a population down here of EVs that pick up the CD63 staining but did not express GFP. And we understand that to be wild type CD63 on some of these EVs. And that's also to be expected given the transfection protocol that's used. So we can be quantitative over any one of these quadrants or really any gate that you like. So what I'm showing you here are the particle concentration versus size for any one of these subpopulations as indicated by the color. Let's look specifically at the double positive population and take this a step further, because the fluorescence that we measure is quantitative and it's in these special units of MESF, right? So we make it very easy for the instrument to be calibrated in terms of MESF units. This unit is a NIST-defined unit, which basically means if you measure something to be 10 PE MESF, then that particle is as bright as 10 free-floating molecules of PE in solution. that's a really, really powerful unit because you can take that unit from one instrument to another. It's translatable, right? And you can compare results. Compare that to your normal flow cytometry data, which is just arbitrary units of scatter or detector voltage. This is a much more powerful unit. So we can also relate that to the physical situation, right? So we look at this population. Remember, this is the double positive population. We see that there's a group of, the majority of the population here has about has brightness of about 3 PE-MESF. So that means the majority of the particles are about as bright as 3 free-floating molecules of PE in solution. And because we know that roughly one antibody has one fluorochrome on it, this we can estimate to be about 3 antibodies per EV. So we can start getting quantitative about the number of epitopes of CD63 that are on the particles in this sample and ask questions like that. So this is a very powerful measurement that way because the units are quantitative. We can say in this example that there are about 3 CD63 molecules on the surface of each EV in this subpopulation of the sample. Okay. So hopefully this example has shown you direct and accurate measurements of EVs. We measure the size and concentration directly, as well as brightness in quantitative measurements. We've quantitated or quantified surface markers on the surface of these EVs. And the instrument's very fast and easy to use. We have videos on our website that will show you, virtual demos of what the workflow looks like. So that's this example. Remember, if you have any questions, please put them in the chat. We can talk about them at the end. So next thing, I would like to take this up a notch and measure fluorescent particles directly in plasma. So again, I'll show you an example that's illustratory, and then after that, we'll do the real full EVs in plasma measurement. In this example, what we did was we took plasma and we serially diluted 100 nanometer fluorescent particles into this plasma. Okay, so we spiked them in at a range of concentrations. Then we measured the samples on the arc. And just let me say something about the sample prep here. So we did immunostaining. That is a no-wash protocol. You just add the antibody, incubate, and then measure the sample on the arc. We're far less susceptible to background fluorescence compared to normal flow cytometer measurements. So there's very little sample prep required. In this case, now we're measuring plasma. We spiked in the fluorescent particles and we diluted tenfold for measurement. Okay, and that's pretty much the extent of the sample measurement or the sample prep. So it's very fast. Measurements themselves take about 5 minutes per sample. So this is one example of what the data looks like, where we've spiked in fluorescent particles, and I'm showing you 2 different size distributions. In blue is the distribution of all the particles in the sample, so that's plasma particles plus the fluorescent ones. And in green is just the size distribution of the fluorescent particles in the sample, so this isolated subpopulation in the mixture. At this spike in concentration is pretty high. We spiked it in at 10 to the 11 particles per mil. What you can see is that the overall size distribution, is dominated by the fluorescent spike in at 100 nanometers at this high concentration of spike in the concentration rate. But as we dilute out the fluorescent particles, so we spike them in at lower and lower concentration, you can see them drop down to the level that they're far below the concentration of the plasma. So same legend here. The blue is all the particles in the sample, and the green one are the fluorescent ones. But in this case, the particles at the 100 nanometer size range are totally dominated by particles in the plasma, and the fluorescent ones are a minority subpopulation. We can look at this data in a few different ways, but the most compelling one is in this bottom right here, where we just report the measured concentration of the fluorescent particles and the plasma across this serial dilution. And you can see that we can accurately quantify the concentration of the fluorescent particles as a subpopulation in the plasma down to about 100-fold lower concentration than the plasma itself. So here we're accurately quantifying a subpopulation of particles using their fluorescence directly in the plasma using the arc. All right, So that's what this example shows. And we think there's utility. We have customers that are interested in applying this technique to measure the fate of nanoparticles directly in complex media. So this is kind of like a PK test for nanomedicines directly in a relevant biofluid. I'll be happy to talk about that more in the Q&A after. It's kind of a different application. Okay, so the final example I'd like to discuss is one in which we did immunostaining of EVs and quantification just directly in the serum. And for this example, what we'll use are these also recombinant EVs from Hansa Biomed. This is a cool company in Estonia. They have recombinant EVs that were grown in HEK cells, and they also express this fusion between CD63 and EGFP. Okay, so very similar to the other sample, different supplier. Okay, and in this experiment, I'll show you measurements of 4 different samples. In blue, the data will be measurements of the unstained EVs by themselves. In green will be the EVs after we spike them into the serum. Then we'll do immunostaining. That'll be shown in red with a PE conjugated anti-CD63, just like we did before. And then as one of the controls, I'll show you serum-only control. So we did the immunostaining, but without the EVs in there at all, right? So we're just checking for non-specific staining of serum particles with this method. So the sample prep, again, is very straightforward. We add the EVs to the serum. If we're going to stain them with the antibody, we just add the antibody, we incubate, and then there's no sample cleanup or anything after this. We just measure them directly on the arc afterwards. Okay, no washing of the free unbound antibody. All right, so let's start with just the size and concentration. So that's what's shown here for every sample. And if you notice that EVs by themselves in blue are the lowest concentration, concentration goes up when you add the serum, right? Because the serum is a higher concentration than the other one. So concentrations are additive, okay? And now let's look at the fluorescence data. So this is showing the brightness of particles detected in our green channel. So here we're detecting the GFP, right? Versus their particle size. And again, this is directly measured. So this is a very heterogeneous sample, right? We've got EVs, we've got antibodies, we've got serum and all the components in the serum all mixed together. And we're able to directly pull out the GFP signals, and I'll show you antibody staining also directly in here, and accurately measure the size of all the different materials in this sample. So what we see is what's expected here. Any sample that does not, any sample that does contain the GFP EVs shows a concentration of particles above our detection level in green, right? And the serum by itself, so there's no GFP EVs in this sample, essentially no positive signals for GFP, right? Only 1% of the particles in the sample are above threshold here, and that's mostly due to where the detection level is set, right? Whereas the sample by themselves is like 30% positive fluorescence. Okay, we can look at the brightness distribution of particles in this sample. And so in this plot, what we're looking at is concentration on the y-axis versus the particle brightness on the x-axis. And essentially the point of showing this is that it says that adding the EVs to the serum does not change the brightness of the GFP, right? So this distribution looks the same whether the particles are by themselves in blue, or whether they're in the serum in green, or whether they've been immunostained with the PE anti-CD63, and that's in red. Again, the concentration of fluorescent particles in the green channel in the serum-only sample is essentially 0, right? It's far below the concentration of the other ones, and this is in the noise, right? All right. So now let's look at the PE channel. So this will be detecting the fluorescent staining using the antibody for CD63. So same plot as what we looked at before on the left here, except now we're looking at the brightness on the y-axis in the yellow channel. So this is just for PE staining, right? And here you can see that the only sample that shows significant amount of fluorescence in the yellow channel are the ones that we stained with the anti-CD63 antibody, right? That makes sense. None of the other ones get the antibody. And our negative control is working here. When we try and immunostain just the serum by itself, we don't get any fluorescence either from this measurement. And we can look at that again in terms of the brightness distribution. So all the samples except for the red one have a very low concentration of fluorescent particles in our PE channel. That's because they didn't pick up the stain at all, right? Finally, one more view of this measurement, like we were looking at before, is this two-dimensional fluorescence cross quadrant plot, where we've got GFP brightness on the y-axis and PE brightness along the x-axis. And it's a similar story to before, which is that the GFP expressing particles all pick up the CD63 antibody. That's expected because they're fused together, right? And that the immunostaining is specific. So nothing in the serum is picking up either fluorescence. You know, there's no GFP in those particles, but there's also no affinity for the antibody in that sample. Okay, so this example has shown that we can do subpopulation analysis directly in complex media, right? This is EVs done immunostaining, phenotyped directly in plasma. It's accurate and quantitative analysis for brightness, size, and concentration. And it's very simple to use. As I mentioned, that we didn't do any sample cleanup of this sample in advance of measurement, and there's no calibration, alignment, or cleaning of the system required. So that's the data that I had prepared for you today. Just to summarize now, so the ARC makes very direct and accurate measurements of three basic properties: particle size, particle concentration, as well as the fluorescence of individual particles. We go down to about 50 nanometers, smallest detection limit for now. And in order to do this analysis, you know, in the results, there are essentially no assumptions required to interpret the data. So the accuracy is independent of refractive index, right? I've shown you that we can accurately measure really complex mixtures of materials, right? And of size, so polydispersity also doesn't matter. It's also fast and easy to use. We talked a little bit about the flow protocols, but also, you know, just operation of the instrument is very easy. We've got a lot more information on our website. Come visit it and learn more. We have videos, short videos showing, you know, demos of the technology, as well as overviews of how it works. We've got application notes, including the data that I showed today, just in more condensed form. And we would love to hear from you either through there or here in the Q&A. Finally, on our website, we also have a library full of webinars available, pre-recorded webinars. So I encourage you to go check them out. The most recent one on the EV topic that we've done was with Dr. Mickey Pentecost from Diadem Therapeutics, who is engineering EVs for drug delivery, essentially. So that's a pretty fun one. He gets into a lot of technology comparison stuff in that presentation. So thank you very much. And at this time, I'd like to open up the Q&A to any questions you might have.
Thanks very much, Dr. Fraikin. There's one question in the Q&A box from Maggie Witek. And the question is, concentration is expressed as number of particles per mil per nanometer. Can you explain this unit again, please?
Yep, thanks, Maggie. That's a very shrewd observation. So if you look at this size distribution here, we've got concentration on the y-axis and particle diameter on the x-axis. And I said that the concentration is measured in terms of particles per mil. It's plotted here in terms of particles per mil per nanometer. And the way to think about that unit is just that if you measure 10 to the 9, for example, at 65 nanometers, then that means that there are 10 to the 9 particles per mil in a 1 nanometer bin centered at 65 nanometers. Okay, so essentially what we do is, the reason we display this unit is so that it's agnostic to the binning, you know, how wide you space these points, because otherwise you can pretty drastically change the appearance of the distribution just by how you bin the size binning, so of the histogram. So it's particles per mil per nanometer. If you want to know the concentration, say between 150 and 200 nanometers, you just integrate this curve from 150 to 200, and then that tells you particles per mil in that size range. This is really a fundamental unit that makes it more translatable from one measurement to another, and less susceptible to user choices about binning. You can rebin the data as you like and display the data as you like in our software. And you can also measure these concentration ranges or concentrations over any range just by click and drag with the mouse or in some canned reports that we have available in the software. So it's very straightforward. Yep. No problem, Maggie. Thanks for the good question. Okay. All right. So there's a question in the Q&A. You mentioned in the beginning of controlling the particles size to enter. So I'm assuming that's about the filters. And then the question is, what is the minimum size that can be entered, and how exactly do you control to enter one size at a time? Okay, so a couple of parts to that question. Let me just go back up to the diagram because I think it's helpful. All right, so we control the maximum size of particle that can enter using this filter structure. So this is shown here. What it is a network of small channels, and all the particles have to pass through one of these small channels before they can reach to the sensing constriction, right? This is where we do the actual measurement, but we don't want big particles getting here because then they will block the constriction. That's what happens in the Coulter counter. That's what happens in other implementations of RPS. So our innovation has been to build in this filter structure that just sets an upper limit to the size of particles that can be measured in a given cartridge. We have a range of different cartridge sizes to span the range from 50 nanometers up to 10 microns at the top end for our technology. And so each one has a range over which it measures particle sizes. Your question also mentioned the minimum size that can be entered. So we don't control the size, the small particles. Small particles can flow through as long as they pass through here, no problem. Our detection limit at the small end is determined by electrical noise. And so that's well defined. It's not sample dependent. And what that means is that on a given cartridge, we can tell you the range of particles that you're measuring. Anything that's smaller than the detection limit of the particles, so anything below 50 nanometers, for example, in our smallest cartridges, just don't generate a signal that's appreciable enough to be detected. So they can flow through here all day and you won't see them. So really we just measure on the stated range of particle sizes given for a given cartridge. So hopefully that answered your question. There's a question here. If I need to check some protein markers and if they express on the surface and inside the EV membrane, can we differentiate both markers? Okay, that's a good question. So we have means of making internal nucleic acids fluorescent, but if you're using an immunostaining approach, the antibodies are not going to penetrate the EV. So any fluorescence that you get from immunostaining is probably going to be externally labeled, or externally bound proteins. I don't think you'd have any cross signal or any like confusing signal from antibodies going inside and finding the target that way. In the LNP world, which is another part of our business, we use intercalating dyes to light up the nucleic acids inside nanoparticles. And so in that case, then we're labeling the inside part, and that's just a different kind of approach. We could actually, I believe we've done this for EVs also, detected internal nucleic acids using that same idea. I didn't show the data here today. That'll be for a future webinar. So hopefully that answers your question. Okay, there's a question about does particle compressibility affect size assessment? I don't believe so. It'd be very hard to check, I think, probably. But one thing to keep in mind is that all the flow through the cartridge is laminar flow. And so there's really not a lot of compression of the fluid. So I'm not sure how you would compress a particle except if it's, maybe getting squeezed as it goes through the constriction, but that would not be a high quality measurement, I don't think. So I think to the extent that we understand how the system works, particle compressibility does not impact the measurement at all. Okay, there's a question about double labeling particles should be showing in yellow color instead of red in fourth quadrant if both single labeled is green and red. Yeah, so the colors in the plots I showed are really just an artistic impression. They're just chosen for aesthetics, so they're not the actual colors of the particles that are being detected. Okay. There's another question. I just received one batch of new cartridges recently. Do I need to calibrate them? No, the cartridges are pre-calibrated, so you don't have to do any calibration on site. Okay. And then looks like last question, or two more questions. Can you permeabilize EVs, much like cells, to allow antibody entry? That's a good question. And if you have ideas for or working protocols for that, Stanley, we'd love to hear from you about that. So shoot us an email and we can talk about doing some sample measurements or something to answer that question. That would be a really cool experiment. So reach out to us, please, about that. Okay, and then last question that I see in the chat right now is how do the fluorescence detection limits of ARC compare to those of other nanoscale flow cytometers? What is the minimum number of surface proteins that can be detected on an EV surface? Okay, that's a good question. So I don't have the data on this presentation, but it's available on our website where we used cross-calibrated beads to assess the detection limit of the ARC. They were assigned a brightness using MESF standards that are NIST traceable. And then we measured our detection limit, where the electrical floor, or where the electrical noise floor is. And so what we found when we did that was that our detection limit for FITC was in units of FITC MESF was about 20, 25. And for PE, obviously PE is a much brighter fluorochrome. So it's in the range of a couple of fluorochromes. So, and I think the data that I showed today showed like 2.5 PE as the detection limit. Okay, one more question. Can I use unprocessed plasma and serum? I mean, essentially the data that I showed today was that. We remove the cells first though, but that's inherent to plasma or serum. And we do dilute it in PBS, I think tenfold usually for measurement. So hopefully that answers your question, but otherwise there's no significant sample cleanup to be required for sample measurement. Okay. Any other questions at this point? If not, I think we've reached the end of the time.
Thank you, ladies and gentlemen. Thank you, Dr. Fraikin, for a great talk, ladies and gentlemen. Spectradyne will make the recording available on their website and send out a link. If there's no further questions, we'd like to thank you all, and we look forward to the next webinar by Dr. Fraikin. Thank you, everybody.
Thank you very much, everybody. I appreciate your time today.
Thank you.